am Dr. Vaibhav Wani, Assistant Professor in the Department of Guna, College of Ayurveda and Research Center, Nigri. Today I am going to demonstrate one of the type of the microscope. It is binocular. This is microscope, and today we are going to start from the main parts in this binocular microscope. This is the two eyepieces, eyepiece lenses, that is ocular lens. This is one, this is two. Then here is the diopter adjustment system is there. This is head of the microscope. This is head. This is called as nose piece. Then these are three objective lenses of different power: 160x, 100x and 40x.
10x.
And 10x is also there. Then stage layer clip. These are the stage clips, and the glass slide mounted on it. This is light illuminating. So here is the illuminator, that is light source, and here is the switch by which we can on off. This is base. Now we see the principle. What is the principle? Principle is in a simple light microscope, a thin specimen containing a slide is placed on the microscope stage. A beam of light is passed through the condenser to the specimen. The light transmitted from the specimen enters the objective lens. While passing through the objectives, the transmitted rays are spread so that they appear to come from the bigger objects. The light is then focused on the eyepiece lenses. This lens further magnifies the pre-magnified image coming from the objectives. And then finally a highly magnified image can be observed from these both eyepiece lenses. Then we can see the structural parts of a microscope and their different functions. There are three structural parts of the microscope, that is head, arm and base. The head, what is the head? The head is a cylindrical metallic tube that holds the eyepiece lens at one end and connects to the nose piece at the other end. It is also called a body tube or eyepiece tube. Further, it connects the eyepiece lens to the objective lens. The light coming from objectives will bend inside this tube in binocular microscopes. Then they are adjustable so that the viewer can adjust the eyepiece for maximum visualization. Here is the adjustment. These are the fine and micro adjustments. Then arm. What is the arm? This is the part connecting from objectives from connecting the base to the head and the eyepiece tube to the base of the microscope. Okay. Then it supports the head of the microscope and is also used when carrying the microscope. This is the arm. Okay. Then some high quality microscopes have an articulated arm with more than one joint, allowing more movement of the microscope head for better viewing. Then base. What is the base? The base is the lowermost part of the microscope that supports the entire structure. Also, it provides stability for the microscope. Illuminator. This is the illuminator. Illuminator's light switches and electric wiring systems are fitted in the base. So this is the equipped with wiring for light illuminating sources. Optical parts of the microscope and their functions we are going to study. Now the optical parts of the microscope are used to view, magnify and produce an image from specimen placed on slide. We have placed here a leaf of guduchi plant. Then the eyepiece. What is the eyepiece? Eyepiece is the ocular lens. This is the eyepiece closest the viewer's eye. Hence it is called eyepiece. Viewer is placed where both eyes on this eyepieces. They are located at the top of the microscope. This part is used to look at the specimen. These lenses come in different magnification powers from 5x to 30x. But the most common ocular lenses are of 10x or 15x magnification. They magnify the image for second source of time. Then eyepiece tube. This is the eyepiece tube. Its eyepiece holder. It carries the eyepiece just above the objective lens. In some microscopes such as binoculars, the eyepiece tube is flexible and can be rotated for maximum visualization for variance of distance. For monocular microscopes, they are non-flexible. Then diopter adjustment. Diopter adjustment is a control knob present only in the binocular microscope. This is the binocular microscope. Hence, diopter adjustment is also there. It is used to correct any difference in vision and compensate for the differences in vision between viewers' two eyes. Then nose piece. This is the nose piece. Nose piece is a movable circular structure. You can see this is movable circular structure. This is called nose piece. It is also called revolving turret. It is connected to the body tube and lies just above the stage. It can be rotated clockwise or counterclockwise to increase or decrease the magnification. The change in magnification results due to change in the objective lens. Objective lenses. These are objective lenses. The objective lens is the lens that is closest to the specimen. This is the objective lens closest to the specimen on glass slide. They are fitted on the nose piece. A standard microscope has 3 to 4 objective lenses of different magnifying powers, that is 4x, 10x, 40x and 100x. The objective lenses first receive the light transmitted from the specimen and magnify the image for the first time. Objective lenses are color coded and are different sizes. Size and color depend on the power of the lens. Then the smallest lens is of the lowest power and gradually the longest will be of highest power. The high power lenses, that is 40x and 100x, are retractable. That is, what is retractable? That is, their end can be pushed inward. In most optical microscopes, objective lenses with 100x or more magnifying or more magnification are of oil emulsion type of lens. The adjustment knobs. The adjustment knobs are the control knobs used to focus the microscope on the specimen. These knobs are two types. Fine adjustment, that is, you can see here is fine adjustment, and second is coarse adjustment. This is fine and this is coarse adjustment. What is fine adjustment? Fine adjustment knob is used for fine adjustment. It is smaller knob and is used to move stage up or down very slowly. The stage covers a very small distance on each rotation of the fine adjustment knobs. It is used to sharpen the image. It is mostly used while viewing under high power microscope. Then coarse adjustment knob. Coarse adjustment knob is used for focusing the image under low magnification. It is larger knob and is used to move the stage up or down very rapidly. The stage is raised or lowered rapidly with the help of coarse adjustment knob. This is coarse adjustment knob. Then stage. This is stage. This is the section in which specimen is placed for viewing. They have stage clips that hold the specimen slides in place. Okay. The most common stage is the mechanical stage, which allows the control of the slides by moving slides using the mechanical knobs. These are the mechanical knobs on the stage instead of moving them manually. Then aperture. This is the hole in the microscope stage through which the transmitted light from the source reaches to the stage. Microscope illuminator. This is the microscope illuminator. It is a light source. In some compound microscopes, a mirror which reflects the light from an external source is used. This is the optical microscope. different electric bulbs of low voltages are used as constant light source. This is trinocular. Same is as of binocular. This is the light source connected, and different optical lenses are there. And this is the another third camera of observing. The main difference is it is have a third camera used eyepiece. You can see used for the better magnification.
Camera
For capturing the details and detail zoom magnification of the glass slide material. Thank you.